Okay. So welcome everyone to our expansion user group meeting. We are very lucky today to have Ben joining us from Adelaide. He spent a long time in the US now, and he's coming back to try to again reestablish expansion. So we're very lucky here to hear from him and to be able to learn a lot of things. Unfortunately, my video is sort of not working, so you have to just stick with my voice. And Ben today is going to talk to us about how he used expansion in malaria and parasites. So up to you, Ben.
Well, thank you very much.
Mm-hmm.
Yeah, cheers to the introduction. I just need to minimize these little windows so I can see my screen. Everything working good?
Yeah, that looks good.
Beautiful. All right, I'll use a laser pointer. So yeah, cheers everybody for coming on. And yeah, I'm going to tell you a little bit today about expansion microscopy and malaria parasites, and what I've done over the last few years of my my postdoc studies, and sort of hope to give you a flavor of the area I'm hoping to move into as I now that I'm back here in Australia. So I want to acknowledge that I'm coming to you from Ghana country, the University of Adelaide, but that pretty much all the work that I'm going to present here is done at the Indiana University School of Medicine, and that IU School of Medicine is built on the lands of the Shawnee, Delaware, Miami, and Potawatomi people. So yeah, like I said, today's about malaria and expansion, but I wanted to start by introducing a few really basic kind of biological concepts that sum up part of why I think the biology of parasites in general, but particularly malaria parasites specifically, are really cool. So the first concept is so basic that it's kind of easy to forget about, but most cells do not permanently exist as kind of amorphous blobs. They have highly specialized structures and organizations that enable them to carry out their functions. And form follows function. That phrase that we always hear in structural biology, in my opinion, applies as much to cells as it does for protein structure. So here I have a few examples. Maybe the most extreme example in humans is neurons, with kind of incredibly branched and intricate cell bodies that enable them to interact with a large number of cells and also have like long-range cell-cell interactions. Epithelial cells, in this case from a kidney, which show strong apical and basal polarity, with the apical end containing the microvilli. And a budding yeast, where a site of polarity is established here to enable the formation of a daughter cell. There are countless examples of this across biology, but the key takeaway is that cell structure is specialized for function, and that this typically occurs through the establishment of polarity either prior to differentiation or at a certain life cycle stage for, say, single-celled organisms. Considering the audience, this is going to be a very visual talk. And I wanted to share one of my favorite examples of kind of specialized cell structure. This is a ciliate single-celled organism that kind of lives in marine and freshwater environments called Euploides. It's about 75 micron in length, and it contains all these kind of hair-like structures or bristle structures called cirri on the outside. And that enables them to undergo some kind of pretty cool and interesting movement patterns. I'm not going to talk about Euploides today, but I think this is kind of good idea, a good kind of reiteration of why specialized structure relates to function. These really kind of interesting and intricate parasites have these really cool movements that allow them to, sorry, they're not parasites, single-celled organisms. I'm getting ahead of myself. The other concept that ties into this is, of course, that cells undergo really dramatic reorganizations to undergo mitosis and cytokinesis. So a daughter cell will inherit the right amount of all the different components of the cell. Most cells that have too many or too few of any particular organelle will likely die or have some serious dysfunction. But I work on malaria parasites, so why are these kind of cell biology concepts interesting to look at through the lens of parasite biology? And so here we have a stage of the parasite life cycle called an oocyst. This grows on the mosquito midgut wall. And right now, this oocyst is really kind of a bag of bits. It contains hundreds or thousands of genome copies, although far fewer nuclei, which is an interesting thing. A mitochondria that'll get broken up into hundreds of pieces, and many of the other ingredients that you need to build a parasite that can invade a host cell, in this case, a mosquito salivary gland, and then eventually a human hepatocyte. So these invasive stages are typically called, collectively are called zooites. And from this kind of parasite soup emerges this, a hyperpolarized sporozoite in this case, that shows extraordinary apical basal polarity with all the organelles that are required for host cell invasion down here at the apical end of the parasites. And so malaria parasites undergo a dramatic morphological change like this 4 times during their life cycle. And for each of those times, they must enter host cells. But the extraordinary thing to me is that in each of those instances is done differently every time. So here we have a blood stage malaria parasite, which is called a ring stage parasite, inside a red blood cell. And at this point, it kind of undergoes like an amoeboid motion floating around inside the red blood cell. But in less than 48 hours, it forms around 20 of these highly polarized merozytes. Merozytes are also made in hepatocytes, where a single liver stage parasite can give rise to hundreds of merozytes. And then, in my opinion, the most extreme example of this is in the oocyst, as I mentioned, where in only a few days, a seemingly featureless oocyst can give rise to thousands of these highly polarized sporozoites. So when you think about what I've just said, malaria parasites form dozens to thousands of highly structured daughter cells ready to invade new host cells. And it kind of marries up these two biological concepts, firstly, of structure equals function, and then cell reorganization for mitosis and division. And thinking about this, I'm really interested in kind of three questions. So firstly, how do zooites determine their structure that they need to invade cells? How do parasites get ready for division, especially considering that they're making tens to thousands at the same time? And both of these are kind of bridged by the very broad question that I often think about of overall, how do you build a malaria parasite? And so I'll start off by explaining why this is challenging. And as cell biologists, our primary experimental tool is light microscopy. But that leads us to one of the major challenges of studying cell biology of malaria parasites, which is that they're really small. So here we have some sort of to scale electron microscopy images of a fission yeast, an E. coli, and then over on the right, we have a single malaria parasite, in this case, a merozyte. These parasites can be about as small as a single bacterium, but we're interested in cell biology. We need subcellular details. And so one of the organelles that I'm going to talk about today are these guys outlined in red. They're called the rhoptries. They're a major secretory organelle that the parasite uses to invade host cells. And so they're taking up only a fraction of an already small parasite. And considering that they can be only a few hundred nanometers long, you can really imagine the difficulty of studying rhoptry biology and kind of dynamic changes over time using conventional light microscopy. So how do we do this? And I guess it's probably no secret considering the forum. The answer is expansion microscopy. So on the left here, we have a blood stage malaria parasite stained with a protein density stain NHS ester in grayscale. I'll use this all throughout the talk. We also have tubulin in magenta and a DNA stain in cyan. And in this image here, it's kind of really difficult to work out a lot of subcellular detail. But when we expand this, however, you can immediately appreciate how much more information we can extract from these images. This is a night and day comparison. And as a parasite biologist, I'm kind of legally obligated to show a life cycle at some point. And so this is my malaria parasite life cycle diagram where all the parasite life cycle stages are represented by sort of bright field or similar kind of imaging methods. But this life cycle has got a really substantial facelift in the last few years, with expansion microscopy having been applied to all of these life cycle stages, really dramatically changing what we see in each of them. And I hope you can appreciate how much of a difference this makes in our ability to visualize malaria parasites and therefore, importantly, study their cell biology using light microscopy. So the focus of today will be on some of the life cycle stages in the mosquito. But for kind of up until recently, I'd spent most of my research career studying this blood stage of the life cycle, the asexual blood stage. And that's how we really started using expansion. So I'm going to tell you a little bit about how understanding cell biology of these guys enabled through expansion helped us to move and ask important and interesting questions about other life cycle stages. So to introduce the people who were actually involved with that work, this is all published, so I'm going to try and go through it quickly. But this was a kind of an exciting collaborative project where we brought together a team, geographically spread team, where everyone brought their own areas of biological expertise. And in a basic sense, this project was enabled just by some kind of methodological changes that I made to existing expansion protocols that made it substantially more practical to harvest multiple stages of the parasite life cycle while they're inside red blood cells. So here again, we have unexpanded and to scale comparisons of expanded parasites. I haven't included the unexpanded free merozoite because it's almost too small to bother. So what we did was to image expanded parasites across the entire intra-erythrocytic developmental cycle using markers for all of the 14, I think it's 14, cellular structures listed on the right here. And again, this is published. I won't spend too much time on it. But when you're thinking, when we were thinking of the question that I asked before, how do you build a malaria parasite? From this study, there was something that we just kept coming back to and it kept popping up everywhere. So this structure here, which is called the centriolar plaque, this is a malaria parasite equivalent of a centrosome. So the centriolar plaque spans the nuclear envelope, which is sort of roughly indicated in white. And it has an inner region inside the nucleus that makes the microtubules needed for mitosis, and then an outer region that anchors the nucleus to the parasite plasma membrane here in magenta. So we track centriolar plaque dynamics from its biogenesis up through until its disassembly right at the end of zooat formation. And some interesting implications for this were that the formation of the outer part of the centriolar plaque seemed to be the first establishment of polarity within the cell. This was really important for some thinking afterwards. And curiously, when the centriolar plaque was broken down right at the end of the of zooat formation, we think this is probably to enable flexibility in the nuclear envelope. So this guy gets really heavily squeezed as it moves into the host cell. So our thought for the reason it breaks down, sort of hand wavy explanation, is maybe nuclear envelope flexibility. But once the centriolar plaque got built, a whole range of organelles started associating with it. Most importantly, the Golgi, with pretty much all the Golgi-derived structures subsequently forming at the centriolar plaque, including of importance today, the rhoptries. And kind of importantly and interestingly, all of these organelles coordinated their duplication and their inheritance with centriolar plaque duplication and separation during mitosis. And then finally, we showed that these two branched organelles, a parasite-specific organelle called the apicoplast, and then the mitochondrion, attach a branch to the outer centriolar plaque of each forming merozoite. And we think that this enables kind of appropriate distribution, fission, and then ultimately inheritance of these structures during cytokinesis. So reflecting on that, we had a few kind of simple questions that have driven my science over the last couple of years. And firstly is the centriolar plaque the major regulator of polarity establishment in blood stage parasites? With new functional data, I think the answer to that now is a fairly convincing yes. Importantly, what proteins control that initial establishment of polarity to make it all possible? And finally, what kind of got me onto the track of what I'm going to talk about today is this the same in the other stages of the life cycle, in the other parts of zooat formation, or even broader, is it's the same kind of concept at play in other parasites? So this gets me onto my work in mosquito stage parasites, and I and Sabrina, who was my postdoc advisor in Indiana, didn't really have experience with mosquito stage parasites. And so we had to build a fantastic team of collaborators, as you can see here. And this project kind of funnily kicked off when Alexis gave a seminar at Indiana University. And Alexis and Sabrina got talking and were like, hey, how about you see what expanded mosquito stage parasites look like? And what started out as a pretty kind of off-the-cuff idea developed into a fully fledged project. But I would say really this project got off the ground quite late last year when I went over to the NIH in just outside of Washington, DC, to work with Tiago and Joel, who are experts in parasite vector interactions. So this was necessary information for me as someone who was going in without any previous mosquito experience. And so I'll quickly run through how we prepare the mosquito tissues for expansion, because everything from now on will be using this technique. And we start by dissecting out either the salivary glands or the midguts from these mosquitoes. For fixing them, we just fixed in 4% PFA. And then here it says to kind of concentrate tissues, and here we do everything in solution, which is a little bit different to a lot of the other samples that I prepped. And this is basically because these guys will not stick to glass, which made it a bit of a pain in the ass. So we kind of worked out a difficult but working way to get around this, where basically we just kind of manipulate liquid volume a little bit to kind of concentrate and space them out, and then eventually turn them into a hydrogel. So it's not perfect, but it's good enough for what we need to do. Yeah, once that's done, we do the pre-denaturation step to detach our gels from our cover slips. And at this stage, quite nicely, we can actually see the mosquito tissues in the gel. So I typically cut out one each and denature them individually. The reason that I usually do one each is because if one of these guys is highly parasitized, it would take me ages to image all the parasites that are in it. So it would, from a practical perspective, be a waste if I had a gel that had 20 salivary glands in it, because I would never be able to image all the parasites inside. So yeah, from here we stain our gel and prepare them for imaging. And then all the images I'm going to present today were taken on an LSN 900 with AreaScan 2. That was just down the corridor from us in Indiana. So I had unfettered access to that microscope, which was great. And as you can imagine, this process, doing it all the time, is a bit of a pain. And so the way that I tried to sort of workflow this and increase the throughput best was by getting heaps of tissues, heaps of salivary glands or midguts, putting them all into gels at once, doing all up to this step, and then basically cryopreserving a bunch of them after this first expansion. That way we've got a big bank that I can keep going back to and staining when needed. So what does this look like? So on the top here, we have an unexpanded, heavily infected midgut, again with a grayscale representing protein density, light blue representing DNA. And I want you to pay attention to the scale bar now as I bring in the same midgut following expansion. So as we can see, the same midgut is now dramatically larger. At this magnification, we can see some improvement. But really the beauty of this technique comes when we're looking at a single oocyst. So I'm going to zoom in on this region, this green area here. We're using the same microscope and the same objective. We're imaging the same oocyst before and after expansion. And now you can see a really dramatic difference. And importantly for us, if we zoom in a little bit further, we can see a bunch of the organelles in the heads of these forming sporozoites. So this was, it's clear the difference between them, and the amount of detail that you can extract from this versus this is massively different. We did the same thing with salivary glands. So on the left here, we have an unexpanded salivary gland. And then if I put that to scale next to an expanded salivary gland, it looks like this. Salivary glands, while we can image the same one before and after expansion, like I did with the midgut, each of these lobes of the salivary glands moves independently. And so even if it is actually the same salivary gland, it looks quite different before and after expansion, which is a bit upsetting, but what can you do? And today I'm kind of hoping I can convince you of the utility of using expansion to understand parasite biology and also thinking about how you can maybe apply this to the system that you're studying too. And so, as I mentioned at the start, on the whole, I'm interested in how you build a malaria parasite. And so today I've used this technique that we developed for mosquito expansion to start looking at things that include oocyst mitosis, organelle fission inheritance, biogenesis of the rhoptries, and then salivary gland invasion. But it's these two on the right, rhoptry biogenesis and salivary gland invasion, that I'm going to talk about today, starting with rhoptry biogenesis. And so many of you not be parasitologists, I recognize that. And so we'll just start with a tiny primer on sporozoite formation before I get into rhoptry biogenesis. So sporozoites develop, so this is in the midgut wall of the mosquito, and they start to undergo rounds of mitosis and nuclear divisions. And following this, the parasite will form a structure called sporoblast, which are basically like groups of what will eventually become forming sporozoites. Then they'll start to establish an apical polarity in the oocyst before forming sporozoites, those invasive stages around this apical polarity. And once they're fully mature, the sporozoites will egress from the oocyst into the hemocoel circulatory system, effectively, of the mosquito. So if we want to investigate rhoptry biogenesis and sporozoites, well, we need to ask, by going back through the literature, well, what do we know about rhoptry biogenesis and sporozoites already? Short answer is not a huge amount. We do know that they're made from Golgi-derived cargo that move as little vesicles along this rootlet fiber, which is connects the nucleus of the forming sporozoite, or centriolar plaque of the forming sporozoite, to the apical polar ring, which is essentially the leading apical end of this forming parasite. And in a more recent FibSem study, the authors described two kind of rhoptry-like organelles. The more electron-dense one, they just termed true rhoptries. And the less electron-dense one, they gave the rather long name of alternative or immature organelles that are distinguishable from the mature rhoptry, or AOR for much shorter. And I'm going to talk about the association between these two things, the rhoptries and the AOR, a little bit later. So if I'm going to start by describing the process of rhoptry biogenesis in blood stage parasites, this is what we found out from that study that I mentioned before. And in that way, we kind of have a baseline, so to speak, that we can compare what we see in mosquito stages too. So the first thing to note here is that we use the parasites progress through cytokinesis as a biological clock. So different parasite stages develop over dramatically different periods of actual time. And so using a biological readout like progress through segmentation gives a frame of reference to be able to compare different stages. So in blood stage parasites, soon after they establish their apical polarity, but before they've started cytokinesis, the parasites start to build their rhoptries. And we also know that rhoptries are duplicated during mitosis and then segregated following nuclear division, and that this repeats a few times, each time ending up with a nucleus having a corresponding set of rhoptries. After a couple of rounds of mitosis, cytokinesis will start and mitosis will finish. And by early to mid cytokinesis, the rhoptries appear to be fully morphologically developed, with distinguishable neck in yellow here and bulb in magenta region. So neck and bulb, it's like a kind of a club shape. And yeah, so after that, basically the rhoptries are morphologically mature. But the key thing basically to take away is that rhoptry biogenesis starts before cytokinesis. Rhoptries duplicate and separate with rounds of mitosis and nuclear division. And the rhoptry is morphologically complete or morphologically mature soon after cytokinesis starts. And so to ask similar kinds of questions based on this about sporozoite rhoptry biogenesis, firstly and pretty simply, when does rhoptry biogenesis begin and end? And how similar is this timeline to what we see in blood stage parasites? Related to that AOR structure before, it had previously been suggested that maybe it's a vehicle for rhoptry biogenesis. So does this structure carry rhoptry cargo and bind to already existing rhoptries? And finally, and probably most importantly, what proteins control rhoptry biogenesis and sporocytes? And I hope I'll give you a little bit of insight into all of these today. Oh, pardon me. So one of the things we noticed very early on was that oocysts, even from the same mosquito midgut on the same day post-infection, can be at dramatically different stages of their sporocyte development. So to compare when certain cell biology events are occurring between different oocysts, we decided to establish a biological clock. And like I mentioned before, we chose progress through cytokinesis or segmentation, as this is something we could easily see, and it's also what we'd use for blood stage parasites. So firstly, just to run you through that, we start off with oocysts that have not started making sporocytes yet. They're basically a bag of mitotic nuclei. Then using tubulin antibodies, we can see the establishment of apical polarity, where the sporocytes that will form around with formation of that structure I told you before at the apical end of the sporocyte called the apical polar ring. Next, we can start to see recognizable heads of sporocytes and the basal complex, which is at the leading edge of those forming heads. And from here, we can start to see the basal complex constricting the nucleus. And at this point, our nuclei have finished mitosis. After this, the basal complex is past the nucleus, and they really start to look like recognizable sporocytes. Finally, we have sporocytes that appear to be morphologically mature, but they're actually still attached to this central structure, sporoblast. Subsequently, however, they will detach from that sporoblast. And so we use basically these 7 steps as our biological clock for tracking rhoptry biogenesis. And so if we want to investigate rhoptry biogenesis and sporocytes, we need to image our parasites from before we can see rhoptries until we have fully mature ones. So using that biological clock, I'm going to show you a series of images that have been arranged in essentially life cycle stage order. So here we have this rather confusing looking structure that if we zoom in, we think that this is the establishment of polarity within the oocyst. Magenta is a marker for the subfollicular microtubules, which form the apical polar ring. And additionally, we can see this rootlet fiber connecting to the centriolar plaque in the nucleus that I mentioned before. So this stage, fairly clear, at least by protein density, that we can't see any rhoptries. Moving into a parasite that's a little bit older, we start to see some recognizable structures. If I zoom in on this forming head here, we can clearly see that apical polar ring now. And we think that this structure here is an AOR and not a true rhoptry. So this is early rhoptry development. Looking at an again older parasite, things start to get more interesting. Firstly, if we zoom in, we can now see distinct rhoptries and AORs. And we see these in a few different kind of arrangements. Firstly, them just next to each other like this, or a really kind of rhoptry and AOR. Then a really kind of intricate interaction between the two where it appears if the AOR is directly docking to the rhoptry, like this or like this. And then another common one we see, albeit this is in a different parasite, is this sort of wishbone arrangement where the rhoptry appears to kind of fork around the AOR. Don't know the significance of this, but you see it all the time. And from here, it kind of increases in complexity to the point where at late cytokinesis, we often have multiple rhoptries, multiple AORs. And this to me is pretty surprising because blood stage parasites finish rhoptry biogenesis not long after the start of cytokinesis. Well, based on the presence of the AORs and what I will tell you eventually is not the final number of rhoptries, our interpretation is that rhoptry biogenesis continues until late cytokinesis in oocysts. Finally, moving into an oocyst that appears to have finished cytokinesis, we see some quite striking differences. Firstly, we don't see any AORs anymore, which we think indicates that rhoptry biogenesis is finished. And secondly, we have a whole bunch of long, thin rhoptries that don't appear to be connected to each other. So putting it together, we made a kind of comparative timeline between blood stage and sporocyte rhoptry development. And overall, there are quite a number of differences, but I'll just quickly summarize what I think are the most significant ones. Firstly, if you recall the three things I mentioned about timing of blood stage rhoptry biogenesis earlier, in sporocytes, all 3 of these are different. Rhoptries aren't visible until well after cytokinesis has started. We find no evidence of rhoptry duplication is coupled to mitosis and nuclear division. And I didn't show it today, but I'm also working on oocysts. And it seems that rhoptry biogenesis doesn't really start until oocysts is practically finished. And then finally, that rhoptry biogenesis continues essentially until cytokinesis is completed. So these two timelines between different stages of the life cycle are totally different from each other. All right, so that's it for rhoptry biogenesis. But our oocyst sporozoites, once they're fully mature, they break out of the mosquito midgut into the hemocoel of the mosquito, where they migrate to the salivary glands. Here, they'll cross a basal lamina and invade a salivary gland epithelial cell before invading the salivary gland secretory cavity. The secretory cavity is actually where the saliva is stored for the mosquito. And this led us to ask a couple of questions. Firstly, a question that hadn't been answered for complicated reasons, which is, how many rhoptries does a sporozoite even have? And then once we work that out, does this number change during salivary gland invasion? And so what was cool about expansion is that we could differentiate sporozoites in each of these stages. And so we imaged expanded sporozoites and then counted their rhoptries. And we first started with oocyst sporozoites, where we found that on average, oocyst sporozoites had 4 rhoptries, with a little bit of variance, but mostly 4 rhoptries. If we took hemocoel sporozoites, which are basically kind of migratory in the process of getting to the salivary glands, we see essentially the same thing, an average of 4 rhoptries. By contrast, when we look at sporozoites inside the salivary gland epithelial cell, they only have 2 rhoptries. Same goes for those that are in the secretory cavity. And this led us to think that during salivary gland epithelial cell invasion, you actually use up a pair of rhoptries. So basically, you go on average from 4 to 2 based on whether or not you've invaded the salivary gland. And that those 2 rhoptries are used up, so to speak. If we took a little bit of a closer look, what we actually saw was something I think is a bit more interesting, where in a mature midgut sporozoite, we tended to see kind of 2 pairs of rhoptries, one at the apical ends, which are in turquoise, and another kind of further recessed into the cytoplasm. And when we measured these 2 pairs, we found that the ones at the apical end were effectively the same length, but by contrast, the other pair had one rhoptry that was about 40% longer than the other one. And because of this size discrepancy, we decided to call the similar length rhoptry pair as congruent rhoptries, and the different pair as the dimorphic rhoptries or dimorphic pair. But this is in the midgut. What does it look like in the salivary gland? Well, when we looked at salivary gland sporozoites, we saw something very striking, which is that we know that they only have the 2 rhoptries, they only have a single pair. It's now typically at the apical end, but when we measure them, the longest one is again 40% longer. And this led us to conclude that this rhoptry pair is likely the dimorphic pair that we had measured in the midgut sporozoites. So putting this together, we think that in midgut sporozoites, we typically have 2 pairs of rhoptries that are morphologically distinguishable, the congruent rhoptries and the dimorphic rhoptries. And upon salivary gland invasion, however, we're left with only the dimorphic rhoptries. And this leads us to think that the congruent rhoptries are likely used for salivary gland invasion, and by extension, it suggests that the dimorphic rhoptries are probably used for hepatocyte invasion. And so to this point, we've used expansion microscopy on wild-type parasites to infer what a timeline for rhoptry biogenesis looks like and what happens to them during salivary gland invasion. But can we look at transgenic parasites with expansion to characterize proteins that control rhoptry biology? There was one problem here, which was that there was no protein we could use as an obvious proof of concept, because no proteins had previously been shown to play a role in sporozoite rhoptry biogenesis. And so now we have time for a bit of serendipity. So we had no obvious candidate for a protein involved in rhoptry biogenesis in sporozoites, but David here was working on this protein called RHON11, and he came to our lab in Indiana to learn expansion from me, and he found that when they knocked down RHON11, their merozoites only had 1 rhoptry, which was awesome. It was a really clear phenotype that they couldn't see before they had expansion. And we also knew that RHON11 was expressed in sporozoites, so that kind of made it our main candidate to test out. But from David's work, there were 3 things that I want you to remember. RHON11 caused a rhoptry biogenesis defect. and an invasion defect. This was separate, and unfortunately, these two can't really be disentangled. But that the last remaining rhoptry, this single rhoptry here, was still secretion competent. And in a study a few years ago, did look at RHON11 in Plasmodium berghei, which is a rodent malaria parasite, whereas David's work was with falciparum, a human parasite. And in this study, they tagged RHON11, showing it localized to the rhoptries, and they generated this promoter swap mutant that I've designated as RHON11 low, which dramatically decreased RHON11 protein levels. So these RHON11 low parasites did not have altered sporocyte production, but those sporocytes that were produced couldn't invade either salivary glands or hepatocytes. And so knowing what we did about the falciparum RHON11 merocyte phenotype, we got our hands on these berghei RHON11 low parasites, and we're curious whether these sporocytes also had a reduced number of rhoptries. And also we wanted to know kind of when temporally the rhoptrid biogenesis defect, if they had one, when it would occur, if it was the same as in sporocytes. So first, the same as in merocytes. So looking at first mature midgut sporocytes in our RHON11 controls, we saw an average of 4 rhoptries, as we expected, but in the RHON11 low parasites, we very consistently saw these sporocytes only having 2 rhoptries, mimicking the merocyte phenotype. And even though the previous study on RHON11 found that they were largely incapable of invading salivary glands, when we looked at salivary glands, we could usually find some parasites. So firstly, here is a control infected salivary gland. Magenta shows a sporocyte surface, and sporocytes are everywhere. With here, this part is a secretory cavity. Antibody hasn't penetrated into there, but you can see the little nuclei. Secretory cavity absolutely full of sporocytes. By contrast, the RHON11 low parasites are really curious. We can definitely see sporocytes, so there's these bits of purple, but their distribution is totally different. Why? Well, in control salivary glands, we always found sporocytes in the secretory cavity. So here is the outline. You can see the outline of a single secretory cavity here, the bit that actually holds the saliva, and there's heaps of sporocytes in it, right? By contrast, in the RHON11 low parasites, we very rarely found them inside the secretory cavity. Instead, almost all of our sporocytes appeared to be physically inside the salivary glands, i.e. they cross the basal lamina, but not inside either the salivary gland epithelial cell or the secretory cavity. So in other words, they could get across the basal lamina, but they can't get intracellular. When we quantified this over lots of salivary glands, you can see that for controls, all the infected salivary glands had parasites in the secretory cavity, while in the RHON11 low parasites, a vast majority of, let's say, pseudo-infected glands didn't have any parasites in the secretory cavity. And one thing that's key about this is that mosquitoes pass sporocytes onto animals through their saliva. So presumably these parasites, even though they're physically in the salivary glands, couldn't be transmitted. And going back to rhoptries, one of the very interesting things we found was that of the sporocytes in the salivary gland, but again, not intracellular, most of them didn't have any rhoptries at all. The newly formed RHON11 low sporocytes in the midgut had 2 rhoptries. And so to me, this says that these sporocytes reach the salivary gland and they're still secreting the remaining rhoptries that they have, leading to these sporocytes that have no rhoptries. So to me, this says that they're likely still secretion competent, and we end up with sporocytes with no rhoptries. So putting this together, we have our kind of congruent rhoptries and our dimorphic rhoptries. In the RHON11 low parasites, however, we only have 2 rhoptries. At this point, however, we can't be sure of which of these combinations we actually have, or if it's a mix of all 3. But whatever the rhoptries that do get made, it seems that they're still capable of secreting if they do get to the secretory cavity, if they do get to the salivary glands, but that they fail secretory cavity invasion, which kind of recapitulates what we see for RHON11 in merocytes. What we don't know at this stage, however, is when the biogenesis defect occurs that leads us to ultimately having 2 rhoptries. In my last few minutes, I'll share a little bit of preliminary data that's ongoing about trying to address this. So firstly, one thing that surprised us, because it doesn't appear to be the case in blood stage parasites, so this is in our RON11 low parasites. Early rhoptry development seems to be pretty normal. These don't appear to be aberrant whatsoever. By mid-rhoptry development, however, we see some pretty striking defects. So we see the formation of these like protein-dense clumps, which are mostly attached to things that are visually identifiable as rhoptries, but sometimes they're also just present on their own, like this. We've seen this across about 20 oocysts and about half a dozen midguts, but I haven't done any kind of detailed quantification yet. It also seems that these structures appear to still be present in mature in mature sporozytes here. So preliminary observation, we're not exactly sure what this means, but I was kind of surprised to see this because for merozytes, they didn't have a visibly obvious rhoptry biogenesis defect other than the fact that they only had one rhoptry. So this might be an area where they're slightly different. Finally, a pleasant surprise. So all of the malaria parasites I showed you in mosquito stages today come from rodent infecting malaria parasite species, so either Plasmodium yoellii or Plasmodium berghii. I've also looked at another Plasmodium species that I didn't show at all today, but a pleasant surprise was that in those three species that I looked at before, the rhoptries are basically sausage shaped. And it's really hard to tell one end from another just looking at the organelle in isolation. But recently, however, we got some Plasmodium falciparum, so the predominant human infecting malaria parasite from Liliana at the Pasteur Institute. And we're both relatively early in their biogenesis and also quite late. We can see relatively easily identifiable rhoptry bulbs and rhoptry necks. So these resemble the merozyte rhoptries a little bit more closely. And so I don't have any follow-up on this yet, but it's an interesting observation, and we're hoping to do a bit more on these in the future, because with the falciparum samples, we have a freezer full of antibodies that work really well. However, with the rodent parasites, most of our antibodies are not cross-reactive. So hopefully, in the not too distant future, all of these expanded images will be having, fluorescent antibodies in addition to just protein density stains. So just to finish with a few key points, we defined a timeline for, firstly, we developed expansion microscopy for mosquito tissues, which was a fun technical task. We defined a timeline for sporozyte rhoptry biogenesis, and we showed that sporozytes have morphologically distinguishable rhoptry pairs with a congruent pair, we think, getting used up during salivary gland invasion. And to us, this suggests that these rhoptry pairs may be specialized for these two different invasion events. As of yet, however, we don't know if they carry fundamentally different cargo, i.e., do they just look different, or are they actively is not the right. Are they qualitatively different? And finally, like it's knocked down in falciparum merozytes, RON11 low sporozytes halves rhoptry number with sort of ongoing work to find out when and how this defect occurs in rhoptry biogenesis. So it just remains me to thank all of the many people who have helped me do this work over the last few years, especially to Sabrina, who was my postdoc advisor and very kind of graciously let me follow, go down a bunch of rabbit holes that kind of had nothing to do with the lab, but ended up being really exciting. And also Danny, who was my PhD supervisor and is now I'm back here in Adelaide working with him. Also to all the collaborators, all the people who helped us with expansion and other things. American Heart Association for paying my salary while I was in the US, and you all for your attention today. And with the time remaining, I'm happy to take any questions.
Thank you, Ben. That was great. We can open the forum for questions if people want to write them down in the chat, or they can unmute themselves. Sorry about that. While we wait for more questions, I have more like a technical question about when you are doing all the expansion of the tissues together, I guess there's some challenge about the orientation. How you get around that?
Challenges of the orientation in what regard do you mean?
I mean,
As in when I was saying I was trying to image the same one before and after expansion?
also like if you are preparing them all together, I'm just trying to get my head around it. Sort of you're making them all together and then you just pick how often you get the right orientation of the organ.
Yeah, it's basically, I mean, that's a major downside about the way that we're preparing them at the moment, is there is still a pretty significant element of luck of the draw. The other thing, so basically what it is, I would put on a few tissues, so a few, say, salivary glands or a few midguts for each thing that I want to image, and just hope that one of them is in a really nice orientation.
Okay.
The other thing that I found as well, because we can't really get these things to stick to the cover slips, the other thing that's quite common is that often our tissue will be floating in the middle of the gel, which is a pain in the ass, right? Because if you have a really short working distance objective, then you can't reach it from either side of the gel.
Yeah.
So this particular problem with the salivary glands, which are very floaty, maybe not surprising considering it contains saliva. But for those guys, I've actually decreased the gel volume quite significantly, which makes the gel much thinner.
Yeah.
So in a practical sense, that's meant that on one of the two sides of the gel, I should, even on like say a 63 or a 40x objective, I should always be able to access enough of the tissue that I can get the images that I need.
Yeah.
Okay, we've got some
more
questions.
Yeah.
Which is great. The
Wanna
first
read them
one
out?
is, what adjustments did you need to make when moving from single cells to tissue samples? Presumably antibody labeling becomes more difficult. So this is completely dependent on what the tissue is. So I have said this as these mosquito tissues, which is completely true. However, we basically use a regular ultrastructure expansion microscopy protocol. And the reason for that is that while these are tissues, we kind of reason that effectively they are bilayers of cells. On either side of that tissue, you only have one epithelial cell in theory. And so thankfully for us, these are tissues that are kind of small enough that we can treat them like they're a monolayer of cells. And the denaturation step of expansion is enough to clear the tissues. This is not the case, however, as we found out when we tried to expand a whole liver lobe from a mouse, and it was a complete disaster. So I think for for tissues that are particularly difficult to optically clear normally, or that are particularly thick, there are always sort of modifications that you have to do there. I think the particular thing with thick tissues to keep in mind is that you can actually get all of the ingredients for your gel into and embedded into the tissue. And what we found when we were trying to do liver sections, which in hindsight were far too thick, was that basically our gel formed around our sample rather than within it. And so that's sort of where that difference comes in. And I think also for tissues that are traditionally difficult to optically clear, just the SDS step of expansion microscopy is not enough to clear the tissue so that you lose all the tissue autofluorescence. And I think that if you were working on those kinds of tissues, kidney, liver, for example, you probably have to do some sort of combination of clearing and expansion to get that to work. But in general, I think if your section is thin enough, this becomes a non-issue. So we've expanded brain slices before, and the area was not a problem so long as they were thin enough. So I think it's a lot about penetration of that gel. There's a question about expansion of red blood cell stages work in relation to hemozoan. Does it expand? The short answer is no. But it is interesting to think about this for your sample, right? So there are certain things that, at least under the conditions that we operate, either won't expand or need a little bit of assistance in getting there. So Paul's referencing in the middle of that blood stage parasite, they've converted a heap of the hemoglobin from the red blood cell into this inert iron crystal called hemozoin. Hemozoin, being a crystal, unsurprisingly, can't expand. And so basically what the particular consideration with blood stage parasites is that they end up with a bit of a void space in the middle where the hemozoin crystal used to be. So it seems as if the digestive vacuole, which is the sort of structure in which it's encased, it seems that that is preserved, the membrane is preserved. However, in the middle, you just have space that has nothing. One consideration, however, is that off-target labeling for antibodies often goes to the sort of void food vacuole space. But no, I don't think that I don't think hemozoin can expand. I think there are ways to digest it, in which case you may get it to expand, but certainly in its native state, it doesn't. There are also some other things that are kind of expansion resistant. So I've done some work with helminths, and nematodes are surrounded by a really highly reduced, as in like disulfide bond. Highly like disulfide bonds cuticle, and they won't expand unless you reduce those disulfide bonds. So there's certain biological things that will prevent something from being expanded or limit how much it can be expanded. But most samples that I've encountered don't have those things in them. We have a question, what sort of system did you use to knock down RON11 and sporocytes? So we actually got the cell line from Takomo's group in Tokyo. And that was a promoter swap mutant. So basically, they took the endogenous RON11 promoter and changed it to a promoter that wasn't of a gene that wasn't expressed during sporocyte stages of the life cycle. Effectively, under that circumstance, you get like no detectable protein or transcript, which is great. And then the controls were, they did that, but then they switched it back to the endogenous RON11 promoter. So that's what the RON11 low. I wanted to call it a knockdown. However, our collaborator was very strong on not calling it a knockdown because it's a promoter swap. So we've gone with the low naming system. Did you leave low RON11 sporocytes for some time? If yes, they may still invade salivary glands. It might suggest an alternative entry into salivary glands. I completely agree with that. So I kind of think that there are two. So the first part is yes. These were parasites that we intentionally gave a little bit more time to get into the salivary gland. But I do think that there are kind of two things going on. And when you look at the distribution of sporocytes inside of a salivary gland, you kind of see two things, right? Every now and again, you'll see like a single secretory cavity that's like 1 or 2 sporocytes in it. And then every now and again, you'll see like a secretory cavity or a region of that salivary gland that has like 1,000 sporocytes in it. And to me, I actually think, again, a bit hand wavy here, but I kind of think that there are certain weak points, perhaps in the basal lamina of the salivary gland, that the sporocytes can exploit. And in the context of the RON11 parasites, it may be that they can only get in those areas where the basal lamina has been disrupted in the first place. This is something that I am not working on because I don't have the mosquito expertise, but certainly Joelle and Tiago are kind of actively pursuing.
Okay, it sounds like those are the ones in the chat. I just have one last question about the cellular structures that you were using, like you were using dyes. Are there particular ones that would not work with expansion?
Yeah, so we use NHS ester a lot. That binds to the amine group of proteins. That works fantastically. All the DNA stains that I've tried have worked. The one RNA stain that I've tried hasn't worked. That was RNA select. However, that could have been a fixation thing. Apparently, it has a preference for methanol fixed samples. Yeah. There are dyes that we've used that surprisingly worked. So like Bodipi ceramide, Bodipi methyl ester. We got them to work by putting them, so they're meant to bind like sphingolipids. We got them to work by putting them on fully expanded gels and never washing them. In theory, lipids are not meant to be preserved chemically using formaldehyde and acrylamide.
Mm.
However, what we think might actually be going on in that instance is that instead of detecting like free lipids, you might be just detecting the distribution of lipidated proteins. So bodepiceramide detects sphingolipids or binds to sphingolipids. So what we may actually be seeing is a distribution of sphingolipidated proteins in that instance.
Yeah.
So yeah, but in a general sense, bodepi works. We haven't had any success with, say, Nile red. What are some other ones? Basically, I think it's almost always it's worth giving it a crack.
Yeah.
For MitoTracker, pretty much all the MitoTrackers are not compatible with expansion except for CM, ROS. I don't know what it stands for, but it's like an orange MitoTracker. And if you put that on live cells and then fix them, it will retain the mitochondrial staining after expansion.
Okay. Thank you. I think that's it. There's no more questions in the chat. Thank you again for a great talk. And I think this will be very useful for everyone attending and also the potential people that will be watching it afterwards. Thank you
Yeah,
very much.
absolutely. I'm always happy to take questions as well. You know, if you want our expansion protocol or if you need help with anything or anything like that, I'm always happy to share our sort of, 1,000-step protocol that's got all the detail that you need that we permanently leave on our bench. So I'm always happy to, as it relates to expansion stuff, I'm always happy to help people out.
Perfect. Thank you very much.
Cheers. Thanks very much for having me.
Okay.